some zooplankton. I'm McKinstry. I think most of the people in here know me. I work at the Science Center. I'm a zooplankton taxonomist and research assistant for Rob Campbell. So a lot of the work that I'm going to talk about today is kind of my portion of his projects. So there's going to be a little bit of physical oceanography, but not too much. We're going to talk more about zooplankton, which is something he doesn't get to talk about all the time. So let's go. Okay, so first off, I'm going to start with an incredibly simple example of a pelagic ecosystem. This is something I usually go over with kids and students and stuff. It's kind of fun, but it's also a really good comparison between terrestrial environment and the pelagic environment, and also just kind of gets our heads wrapped around about the environment we're talking about, because it's very, very different from what we're used to. Then I'm going to go over zooplankton in general, their role in ecosystems and their size. Then I'm going to talk about our research program, the Gulf Watch Alaska, and some of the questions that I want to talk about this evening, how we figured out those questions, our methods, what we found, what the take-home messages are, and then what I want to look at in the future. So first off, we're going to start on the terrestrial environment, and this is my super, super simple example of what's going on land. So we have our sunlight up at the top, and then we have our soil down in the bottom. And one of the big things that we study with our project are nutrients. So nutrients, either the same fertilizers and things that you put in your plants, so I'm specifically talking about phosphates, nitrates, nitrites, silicate, anything like that, iron. So this green blob is just representing nutrients. So in the terrestrial environment, we have our sunlight, we have our soil, we have our nutrients, and together that makes plants happen, basically. So in this case, we're talking about primary productivity. So the organisms that turn sunlight and nutrients basically into biomass available for other organisms, like our bunnies. So our bunnies in this example are our primary consumers. So they're the first step in our food web that's consuming those plants. Above our bunnies, we have predators, especially around here, like our eagles and our foxes. And those are going to consume our primary consumers.
One less bunny.
The same exact thing happens in the ocean. And specifically for this talk, we're talking about the open ocean. So there's no land, there's no hard surfaces that these organisms are ever going to encounter. But we have to take out our microscopes, zoom in quite a bit. And again, we have our nutrients. I just showed you our sunlight. Same exact thing's going to happen, but just on a tiny, tiny microscopic scale. So instead of plants and shrubs and grasses, we have single-celled phytoplankton that make up the primary productivity in the Sound and in the Gulf of Alaska. So this is a cartoon, but it's a really good example of a species of phytoplankton that occurs quite frequently here, where each one of these individual cells is its own little organism, but they like to be attached to each other, and they create a colony like this. So just like our plants attracted bunnies, our phytoplankton is going to attract zooplankton. Now anytime I say zooplankton, I'm not specifically talking about a taxonomic group of things. All of the guys that we talk about today are going to be from a bunch of different phylums, a bunch of different groups of animals. Some of them aren't related whatsoever, other than the fact that they live in the plankton. Okay, so for these guys, these little bunnies here, kind of look like bunnies, they've got nice long antennas. These are our copepods, right? And they live their entire lives feeding on phytoplankton and living in this system. We also have some other weird looking dudes. These guys are called meroplankton because they start out their life cycle as little teeny tiny plankton. So they're small. They can't swim against a current, which is why they're considered plankton. And for this guy up here, this is a crabzoea, so that's basically what a baby crab looks like. That weird alien A looking thing right there, that's going to be a sea urchin one day if it makes it through its life cycle. And then we have lots of ichthyoplankton, or larval fishes that live in the zooplankton as well. So throughout their life cycle, they're going to grow, reproduce, be happy, and our little baby plankton are going to get bigger. They're going to drop out of the pelagic system, and for the most part for these guys, they're going to be recruited into a benthic environment, completely different ecosystem, but not what we're talking about today. Okay, so they're gone. on. Just like our bunnies attracted our predators, our zooplankton's going to attract a ton of different predators in the sound. And so this example here I'm just using is herring. In the springtime, herring are swimming around, finding all of these copepods and zooplankton, telling their friends they need to come and eat as much as they can. And of course, things like forage fishes are going to attract bigger predators too. Like our economically important fish around here, salmon, in this case, I'm using just a Chinook salmon as the example. All right, so that's kind of our orientation for the system that we're going to be talking about today. And that was an incredibly simple explanation of what's going on. Right? This is a super complicated, brain-hurting map of all of the different interactions that they thought were going on maybe probably 10 years ago, but probably needs to be updated. And basically, we just covered what's going down here. So it's incredibly complicated. If anybody ever talks about a food chain, you think of this picture, tell them, it's a food web. There's nothing chainy about it. Okay? So plankton can be incredibly tiny, like the size of viruses and bacteria, or they can be huge organisms, if you think about a jelly the size of this table. But again, they're all plankton, and they're all basically grouped together because they can't swim against currents, right? So they can't swim like a dolphin or a fish. They can't get out of the way of things. They're basically at the whim of what's going on in the ocean. So I'm not going to talk about little teeny tiny things like viruses and bacteria, even though they are really interesting and becoming more and more important as we learn about them. I'm going to be talking about the small things that we can kind of see with the naked eye, but really we need a microscope to look at. So to get us in that space, I have an example because throughout my career, I've had many people say, oh yeah, I was looking at this little organism the other day. It was 250 microns. And I'm like, okay, 250 microns. I have no idea what that size is. All right, that's cool. And then I stop listening, my eyes glaze over. So just to kind of orient you guys for how big things are and what we're going to talk about, I don't want to say something is 4 millimeters. I want to compare it to this thing. So this is an image I took under the microscope, which a lot of these images will be. This is an ordinary, everyday object. I want to see if anyone can guess what it is, and I'll give you a hint. What?
Grain of rice.
Yes! Excellent. That was quick. So this is a grain of rice, and this is how big the guys we're going to talk about are compared to that grain of rice.
Is this long grain, short grain, dry grain?
Jasmine.
Is it, so that'd be about 4 millimeters?
Yes.
Okay, 4 millimeters.
4 or 5.
Got it.
Or 6. Depending. Yeah, I was just going to say, depending if it's organic or GMO. So up here, we have a ton of different groups of zooplankton and a bunch of different taxa. So this guy over here, this is a little larval worm. Over here we have a copepod. Here's a copepod. There's a copepod. These two are copepods as well. This is a pelagic sea snail called a pteropod. This right here, even though it doesn't look like it, it's a larval sea star. It's a bipennarian, so when it grows up, it's going to leave the zooplankton community and settle in the benthic community and actually look like a sea star. This right here, it looks like a clam, but there's actually a little teeny tiny shrimp that lives in there. These are ostracods. We know it's a girl because she has a bunch of eggs. But these are the animals that we're going to be talking about and the size that we're going to be talking about and how big of a role they play in the ecosystem even though they're so tiny. And let's see if I can do it. I have a video of some plankton. That guy squirreling around right there, that was that little ostracod. There's a big amphipod swimming up there. There's 2 amphipods just zooming around. This is a pseudocalanus. This is one of the most abundant, most common species of copepods we have in the Sound. And then here, this is a jellyfish. Whenever I think of jellyfish, I think of these big animals, but really that's about the size that it gets. They can be very, very tiny.
that's what, like a millimeter and a half? Any idea?
We're talking about rice. It's about a half the size of a grain of rice.
Oh, okay, so it's a good size.
Okay, so that's just, again, to give you some background information of what we're talking about today. Now I want to just quickly go over the programs that we're working with and why we're collecting these critters in the first place and what they're hopefully going to tell us. So the research I'm going to present today is part of the Gulf Watch Alaska Long-Term Monitoring Program. And this is a collaborative program between the Science Center and many different principal investigators in the Science Center, different universities, different non-governmental agencies, and some governmental agencies, basically all looking at what's going on in the area affected by the Exxon Valdez oil spill. So this program is trying to assess the recovery of the resources that are affected by the oil spill, specifically herring. Herring play a big role in why we're asking the questions that we ask. And it's also giving us a chance to understand how the ecosystem is changing over time and at 30 years after the oil spill. And what's really cool about this long-term monitoring study is that we're not just seeing effects and worrying about the oil spill anymore. We're actually starting to get an idea of what's going on with climate change and even anomalous climate activity like the blob. I'm going to talk a little bit about the blob and what we've kind of been seeing in the zooplankton and the signatures of the blob's effect on the zooplankton. So there's two questions I want to talk about this evening. One is, how does the zooplankton abundance change throughout the year in Prince William Sound? We think we have a pretty good idea of what's going on, but I just wanted to double check that nothing weird has been happening for the past 9 years. And then, like I said, the second question is, how has the zooplankton community, so the different species that make up those little guys swimming around in there, how has that community changed in relation to the environmental changes we saw during the blob? Practice before, I promise. All right, so here's our study area. With Gulf Watch Alaska, we have 12 stations around the sound in 4 different bays, 2 in Hitchinbrook entrance, 1 down Montague Strait. We have a nice middle kind of representative station in Prince William Sound. This is Rob's autonomous moored profiler, which I'm just going to talk about very briefly in a second. So what we do is we have about 6 to 8 cruises a year that we go out and we hit all 12 of those sites, or 13 if we're working on the AMP, in our research vessel, the New Wave. Many of you have probably seen us on it or seen it in the harbor. And to collect zooplankton, we have a very specific net. Just like you'd be collecting fish, fish need specific nets to catch them in whatever way you're trying to catch them. Same thing with zooplankton. What we're looking at are the zooplankton in the top 50 meters of the water column. So we drop what's called a bongo net down to 50 meters. This right here. It's got 2 nets, so 2 simultaneous samples that it can collect. Drop it down to 50 meters, haul it straight back up, and anything in the path of the mouth of this net is going to be caught and concentrated down here at the cod ends. And that's what Rob is doing right here. So all those little zooplankton guys that you saw were a concentration of what was going on in the entire water column from 0 to 50 meters. Right? Just briefly a little bit about Pamper, or the AMP. I'm not going to get into too many results, but there's some really cool stuff going on with the zooplankton collection with this guy. So the AMP is a moored oceanographic robot, and you can see it right up here. Doesn't look like a robot that you'd think of. There aren't any arms or legs or head or anything. But it's a robot. It has a suite of oceanographic instruments on it, very similar to what we use when we're doing oceanographic measurements on our cruises. So they're looking at things like nutrients, so all those nitrates, phosphates, all that good stuff, looking at salinity, temperature, and now, thanks to NPRB, a couple years ago, Rob was able to collaborate with some scientists at Scripps and build a microscope camera, basically, on the AMP that takes pictures all the way through the water column, so we can get an idea of what's going on with the zooplankton while the zooplankton are there and not killing them like we do when we bring them into the lab. Okay, so this is just a quick overview of how the AMP works. So it's moored all the way to about 720 feet with a train wheel. It has a bunch of line on it, so it just sits at 60 meters. Once its programming tells it to wake up, it goes up through the water column, and while it's going up, it's collecting data, taking pictures. Once it's at the top of the water column, the antenna pops out of the water, sends all the information back to Rob, and then it goes back down and sleeps until its programs tell it to wake up again. So hopefully it doesn't get hit by a ship or anything.
Mhm.
And these are some of the zooplankton images we've been able to collect from that instrument. These are actually not the greatest ones that I've seen so far. And one of the really cool things with these images is we're trying to basically teach a computer system to be able to look at these images and say, hey, this is a copepod, or hey, this is a ctenophore, this is pleuroblachia right here, or hey, that's a larval sea urchin. And it's amazing how difficult it is to teach a computer how to actually look at things and figure out what they are. But that is for another day. We're going to get back to what I do. Instead of looking at images on a computer, I'm looking at everything through the microscope. So once we get our subsample, we take it back to the lab, and I sit there and I figure out who's in there, how many there are, and specifically for copepods, like this guy right here, copepods have a really complicated and interesting life history that also helps us understand different questions about timing, who's an adult, who's not an adult, how many there are. So it's important that we go through and say, hey, that's an adult, that's almost an adult, and also what sex they are. So if we're seeing a heck of a lot of males and not a lot of females, there might be some issues that might be able to answer some questions. Or if we're seeing a lot of females and not many males, that might answer some other questions. So from these data, there are a heck of a lot of variables that we have to sift through to try to figure out what's going on. And there are some pretty hefty multivariate statistics that I've been working on that I'm not going to go over with you guys. If you are so inclined, I'd be more than happy to discuss all this stuff with you after my talk, but I don't want to hurt my brain or your brain trying to explain all these little things. So between 2009 and 2018, so it takes a long time to process these samples, so I'm through the spring of 2018. I've seen about 288 different species and categories of zooplankton in the Sound. Basically, every single phylum from the animal kingdom is represented in Prince William Sound zooplankton. Like I said, not all of them are going to live their entire lives among the zooplankton. There's a nice big list of ichthyoplankton, so these are all the different species of fish that I've seen in the past 9 years, and a heck of a lot of copepods. So let's parse out who we're seeing the most. So copepods, like this big girl right here, these are her eggs, which is why I know it's a girl, make up about 45% of the taxa of the animals that we're seeing in the zooplankton. Next, all the way down at 12%, we're seeing larvaceans, which are basically pelagic sea squirts of a kind. And then these cool little pteropods, which again are pelagic sea snails. We see a few krill larvae, but basically everything after that are just larvae that are going to leave the zooplankton community and go off and become members of a different ecosystem.
Will it go backwards?
Yeah, I'll go backwards. Ah, there we go, different button. So I just wanted to look at a broad overview of abundance. So I'm not worried about species, I'm not worried about life stage, I'm just taking the number of individual zooplankton in each one of those toes, putting them on a plot, and trying to find an average, basically. So that's what's going on here in this plot. So here we have the number of individuals per meter squared. So if you imagine a big hunk of water that's a meter by a meter by a meter, and here on this axis, it's a logarithmic axis because there are so many zooplankton that this graph would make no sense unless you took the log 10 of the zooplankton. So the difference between 1 and 10 here is 10, and then another tick is another order of magnitude, so it gets very dense very quickly. So keep in mind everything that you're seeing down here, that's like 10 zooplankton. All the way up here, though, that's about 10,000. So there's a lot of variability that we see in the sound, and based on season, which was my question that I wanted to answer here. Down here on the x-axis, we have the day of the year. So January 1 is day 1. December 31st is day 365. And then I broke it up by season. So we can see some relatively, I don't want to say normal, because what does normal mean in science? But we can see some basically textbook seasonal patterns of zooplankton abundance overall in the sound. And that helps me because it tells me, one, I did the analysis correctly, and two, super crazy things aren't happening. There are some really interesting things that I'm going to talk about in a minute that are happening, but overall the pattern that we've seen is usually what you would see in a system like Prince William Sound, right? So abundance, total abundance for overall zooplankton peaks about mid-spring to early summer, and then it quickly declines throughout the summer. Sometimes you can see this little blurb of data right here. Every now and again we'll see a fall bloom. Sometimes it's very quick, so we don't catch it. We can only report on things that we're seeing. But this trend follows basically what's going on with the phytoplankton. So in the wintertime, there's not a lot of sunlight, so things can't really photosynthesize. So you don't have a lot of phytoplankton, so you don't have a lot of copepods. In the springtime, as light increases, phytoplankton is going to increase, and zooplankton are going to lag behind but track basically what's going on with the phytoplankton. Once the phytoplankton have used up a heck of a lot of the nutrients during the springtime, a different species of zooplankton, a different group of zooplankton is going to take over, but that's why you see that little bump here that's kind of following the spring phytoplankton bloom. Okay. I wanted to take it a step further and look at who is pushing for these changes, what species or what groups are in charge of these ups and downs. And so I grouped specifically the copepods because, again, they make up 45% of our taxa, and that's what we care about right now. I grouped them between small copepods and large copepods, and we'll talk more about why I did that in a little bit. But first, what we saw is that the small copepod peak in abundance is later in the season, later in the spring and into the summer, than the peak in large copepods. Also, large copepods make up a very small amount, number-wise, of the zooplankton community. But keep in mind, they are way bigger. I tried my best to scale these two. This is a Neocalanus cristatus. It's the largest copepod that we have in the Sound. And it's a really important prey item for fishes. But what's going on here is that large copepods basically are very prominent during the early spring. And during the early spring, when there's all this nutrients around and all of these, all the sunlight around, that's when you're getting a bloom of incredibly large-bodied phytoplankton. So these big disc-shaped diatoms that these big copepods like to eat. Large copepods aren't going to spend a lot of time sifting through super tiny things. Okay? So once those large phytoplankton have taken up all the nutrients in the springtime, that allows for smaller phytoplankton to start taking over, which means that the smaller copepods get to have their day. And that's why they're peaking later in the spring and the early summer than the large copepods are. This is also when a lot of the exciting things are going on during the spring, like the herring spawning and a bunch of other fun spring events. Okay, so I want to quickly just go over the blob, and I have a fun animation. Okay, so this is a map of the world, and we're going to start in 2013. So basically the fall of 2013, and what we're looking at, all of these different colors represent how different the temperature is during that period of time. So warm colors means that the waters are warmer than average. Cool colors means that they might be a little bit cooler than average. But for the most part, the takeaway that I want you guys to pay attention to is 1, the Gulf of Alaska, and 2, what's going on with the yellow and the orange colors. So that's going to be our blob. Okay, so 2013, 2014, it's getting warm. 2015, it's very warm. 2016, warm, warm, warm. Finally, we're in 2017, 2018. Ooh, and that was very abrupt.
Okay, do that again.
That was very fast.
For sure, I thought that was going to just stop at the very end. So that was up until today.
Got it. A little too fast. You got it.
Okay, so like I said, because this long-term monitoring project has been going on for almost 10 years now, or 10 years exactly now, we've been able to capture what was going on before the blob, during the blob, and after the blob in regards to oceanographic, temperature, salinity, all those fun physical characteristics, but also what was going on with the zooplankton and how the zooplankton community reacted to this anomalous event. So here we are. This is shaded for the blob years, so the end of 2013 all the way up to 2016. On our x-axis, we just have the year. The y-axis, this is change in abundance, again, based on a log 10 scale, from the normal. So this line right here, this line is basically that blue curvy line that was just flattened out. And then we looked at all of the different averages and asked how far away from the average are each one of these little signals. So that's what these bars are indicating. So here we see all these bars going up. That means that it was very different from normal, almost over 1. So for this case, since it is over 1, that is an order of magnitude more zooplankton that we were seeing of that particular species. So let's start with just overall zooplankton. We saw a little bit of ups and downs during that blob year, but there was also already variation going on, so that doesn't really answer our question very well, just based on abundance. So the number of guys that are in that system. If you take it a step further and look at different species specifically, things start to get really interesting. There's been others that have looked at the different species of zooplankton in our area off the coast of California, Oregon, Washington. And there's been some very specific copepods that they see down there that we don't normally see up here. So warm water species of copepods. Up here, basically all the ones that I've shown you so far, those are cold water species of copepods. So the guys that we normally see around here. And so this graph right here is answering the question, what was going on during the community? During those warm blob years, we saw a heck of a lot more warm water species that were presumably coming up from the coast of California and Oregon all the way up to the Sound. Comparatively, even though these bars look kind of small. Again, it's an order of magnitude, so there is actually quite a bit going on here. We saw fewer of the normal species that we see here. And that's going to play some very serious roles in our ecosystem when we're talking about how big our copepods are, and nutritionally how valuable they are for the predators in the area. Okay, so most of the years that we saw this, we saw these weirdos, these invaders from the south. I don't want to call them invasive because they can go wherever they want. We basically saw these guys mostly during the fall and the winter. So presumably it's taking a while for all that warm water to travel up from California and Washington state. But we're definitely seeing increased numbers of these small little copepods, and these include things like Calanus pacificus, Mesocalanus tinicornis, a bunch of other Latin names, and this guy right here, Coreicius angelicus, and I'm going to go and talk about him in a little bit because he's very interesting. So we can tell just by the community structure what organisms are indicative of the oceanographic processes going on. So these guys are indicative of warm water masses traveling up to Prince William Sound, possibly from the Washington or Oregon coast. Okay? So climate change and events like the blob, maybe they might create warmer habitat in Prince William Sounds for species like these. So it's clear that even though they're used to warmer waters, they can still live up here. Okay, so just going back, there'll be a bigger picture, but I want to talk about this guy, this Coreicius. Very cool little species of copepod. Warm water, usually doesn't live in the Sound. It has been seen in the Sound since 1989, I believe. But we're seeing them a lot more, and I've been seeing this pattern going on. So this right here is a copepod, here's a copepod, there's a copepod, there's another copepod. This right here is a siphonophore. This is a larvation. This guy, that's another copepod, one of the most common copepods found in the Sound. And this is a larval copepod or krill right here. And I staged this. This was in a sample. I pulled these guys out just for the picture. But what I'm seeing more and more of is these predatory copepods eating or feeding or at least damaging the colder water zooplankton that we're seeing up here, which is really interesting from an ecological standpoint. And this is just kind of to show you how much we've been seeing them in the Sound throughout the years. So here at the very beginning of the study, the really cool thing about this species of copepod is that they're so different from everything else. You can't really, if you have one of these in your sample, you can't really confuse them with any other species. They're very unique. So that way we have a really good, very, very accurate, at least for me, because sometimes I can confuse a few different species, an accurate representation of what's been going on with this community. So in 2010, 2011, we just maybe saw them once. 2012, we didn't see them at all. Starting in 2013, we kind of started seeing them persist over the year. Then 2016, we really saw an increase in these guys. Remember, 2016 was one of those blob years, 2015 as well. Last year, started to see them again, especially in the fall. And then this, in 2018, this is where I've gotten. So far, this is the most that we've seen them during the spring. So questions for me. So shifting gears from...
Have you seen any Para-Euchida, which is the normal carnivorous copepod that we have here?
We do. We don't see them very often. We only see them usually in kind of the more open water areas and usually deep. They're not usually in the
They can eat
top.
fish eggs.
Mm-hmm. They're very cool. They're very cool guys. I could talk about them all night too. So switching from the fact that there's this interesting little predator that we're starting to see more and more of in the zooplankton community, I want to get back to the idea of size. So copepods, you can kind of think of these guys as similar to a bear. So in the springtime, when they're eating all that phytoplankton, they are also accumulating a heck of a lot of fat in their bodies. And that's what these are right here. These are lipid sacs that are just full of fat and oil that they've been able to squeeze out of their phytoplankton food. Basically, these guys, these big copepods, act as sausages for all the fish, including herring and pollock, in the sound in the spring. So it's really important that the abundance of these guys is there when herring are feeding and reproducing. Or herring will end up switching to eating juvenile pink salmon, which is one of the big questions that we have right now, is whether or not there's the impact of a loss of these big players to herring basically eating juvenile pink salmon. When PWSSC decides to release their juvenile pink salmon, they try to time it with the high abundance of these big zooplankton, so that in the hope that herring are going to be eating these big guys instead of going after juvenile pink salmon. Okay? So the question is, are there going to be more predators and less prey? Because if we're getting more and more of these guys, they're smaller, their lipid sacs are much tinier. Again, I tried to scale the size of these copepods to these ones. And if you were a fish, having to run around a room like this to grab individual skittles is going to use up a heck of a lot more energy than just going up to the table and grabbing your big sausages.
Did you see a lot less neocolonists during the blob years?
Kind of. That's a question that I need to look at more before I really want to answer.
Especially the following year, because if they didn't reproduce and go in the diapause the following year, then we would see a decrease probably.
Mm-hmm. Yeah, so those are the results from everything that I've been working on so far. But there's, as you can see, anytime you try to answer one question, you're always going to come up with 5 more questions. So I'd really like to take a look at what's going on with the Coreicius abundance in the sound and see if there's any way to pull out a signal to see if they're having an effect on the community. That's very difficult statistically, because how do you know that you're pulling out the right signal from all the noise? Also, it'd be really interesting to take a look at whether or not the blob is a preview for how climate change is going to affect Prince William Sound and the zooplankton. If these warm anomalous events are going to fundamentally change the way the zooplankton interacts in the sound. And then finally, something I've been reading more and more about and getting more interested in is just how the biodiversity of zooplankton community changes during this program. The more we learn about biodiversity, the more we learn that a very biodiverse system is more resilient to changes. Once you take away that biodiversity, things get very fragile very quickly. But not just in this system, even on terrestrial systems. The more biodiversity you have, the stronger communities that you have. And with that, thank you all for coming. Any questions?
Do you ever, from what I understand, a lot of the copepods go crazy deep in the
Yes.
winter?
They do.
Do you try some deep ones in the deep hole off Naked Island?
We don't have one off of, well, I guess that is
Between
kind of,
Lone and Naked there,
that Prince William Sound, the station that's in the
hole.
very middle,
Oh, yeah.
we do
Big
sample all the way from the bottom to the top
you do.
of the water column, and it's really interesting to see the species that are deeper in the water. So the way their Copepod's life history is set up is, again, they're very similar to bears in some of their life history patterns. In the springtime, those big guys will eat as much food as they can. And the reason why their numbers dip in the summer, it's not because they're dying, it's because they're diving down to depth and basically hibernating from the summer basically until the next spring. And they're living off that big oil lipid sac that they have. So that's why we see them go.
And they reproduce.
Shane.
Well, they're depth.
What's the lifespan like on those larger copepod species that are prevalent around here?
That is a wonderful question. For the most part, it's 2 years. The larger copepods can actually go for the most part, it's 1 year. The larger copepod's life stage can go for 2 years. So the Neocalanus cristatus here, and there are some calanoid copepods in the Bay of Fundy and on the east coast of the Atlantic that live for about 2 years, but they're all pretty big guys.
And is there just one reproductive event in that life cycle, or do they reproduce more than one?
They're spitting out eggs that whole phytoplankton bloom season. So it's not just like they lay eggs and then they're dead. They're just going basically until they run out of energy and die.
It depends on the copepod. Some have external eggs, but the neocalinus is internal.
Okay.
So there's different patterns.
There was a neat picture with the eggs hanging off of them, because I've seen lots of pictures of copepods, but I've never seen one with the eggs. What time of year did you find that one?
She probably would have been from the spring.
From the
Although
spring?
that's pretty lucky because when we're collecting zooplankton, that net damages them
Oh,
and
I
breaks
bet.
them, and
Yeah.
so it was nice
Yeah,
to
that's
see that.
cool to see.
Yeah.
Yeah.
Rachel.
This might be in your presentation, and I'm just slow, but did you look at all at abundance of the warm water species with respect to seasonality in those warm years? Or have you looked at that at all?
Yeah, so that super long colorful graph that I had, the indicator species analysis, is a different analysis, but we were mainly seeing that those warm water species were coming up in the fall,
Okay.
and that they were very indicative of that group. Like from some of the analyses I did, basically all of those species of copepods fell into the same bin, which was pretty definitive for seeing that those guys are only coming up in the fall. We're starting to see more and more of them throughout the year, but as far as their abundance and numbers go, it's usually in the fall. Yep.
Do you see any indication of ocean acidification in your zooplankton, like pteropods or anything?
That would be something really cool to look at, especially with the pteropods. But the problem with our sampling methods is that we do end up breaking a lot of the pteropod shells, so we can't tell whether or not that damages from ocean acidification. We were at AMSS, and there were several talks about ocean acidification and how basically the boundary between acidic ocean and not acidic ocean is rising every year, and I think in some areas it's up to 50 meters, so that would be something really interesting to look at. And Limacina, that species of pteropod, is a really important food item for, I think, salmon and some other guys that basically just target Limacina. So they're an important species to look at, but we can't do it with this program right now.
Okay.
Yep.
So when you say the warm weather species are coming up in the fall of the blob years, so are they reproducing in the south and then coming here?
That is an excellent question, and one that I've been told, no, they're not. But so as I was saying, I look at the life stage of the copepod, and those predaceous copepods, I'm seeing very early life stages, early enough that makes me think that they are reproducing up here. And a lot of the females that I see do have that characteristic egg sac on them, and sometimes they even have little sperm sacs in them too. So they're reproducing. The question is how well they can overwinter, because the water does get cold, and I'm wondering if they are able to overwinter. I think I was seeing several stage between 1 and 6, life stage number 3 copepods in the springtime in the sound.
It's more of a theoretical question, but it looks like there's a lot of different taxonomic divisions
Yes.
of all these things. And in your opinion, because I don't think you've studied it, although you might have, is how quick do these things evolve? In other words, if a warm water species showed up, how many reproduction cycles would it take before it could just live here and reproduce well, just be happy here? Do you think?
In my opinion, probably not in my lifetime or my grandchildren's lifetime or my grandchildren's lifetime. I don't know. I don't know that we know enough about what's called phenotypic plasticity, so just the ability of an individual to deal with environments that it's not used to. Sometimes magically they can, like invasive species that come over here from Asia, and that's not necessarily an evolutionary thing. It's just the fact that that environment is okay. They just haven't had the chance to get into that environment. the really cool thing about Copepod Evolution is that copepods are really hard to fossilize. So there's like 6 copepod fossils in the world. So we really don't know a lot about Copepod evolution. It's very interesting and hard to
Has anybody
study.
tried to just like breed them in a tank and see how quick you can mutate them or anything? Or?
I don't think so. I don't know. Phenotypic plasticity, yeah.
Shot it. When they live for an entire year, it takes a really long time to do that experiment.
I think that'd be like a legacy experiment of many generations of scientists.
Well, people have done that.
Yeah.
Okay.
Beep.
Could you go back to the slide of the whale chasing the herring?
Oh, yeah, that was just the desktop.
I wanna get a photo of that.
Yeah, that picture was taken by Rich Brenner, I think, in,
Oh, it was?
yeah, 20.
I got a copy on my computer, can you send me your email? I'll send you one.
Higher quality, right?
It was, yeah, it was epic photo.
There it is. So this is a humpback cruising through there?
Yeah,
Yeah, that was an A, so it was about to
they
match.
gotta work at it, they don't just swim into their mouth, they run away. Yeah.
The robot, Rob's got out of it. How many times a day is that going sampling or coming up and sending?
Right now, none, because it's not out yet.
Okay.
But when it is out, it does, he has it programmed to set twice a day, once during daylight hours and then once during nighttime.
And how long is it on the surface sending?
Rob, would you answer that?
3 or 4 minutes.
Oh. We're not gonna run into it.
No.
And it is on the charts.
It's on the charts, okay, the charts, so.
Okay. So I'm hearing Son of Blob, is that true? Do we have another blob event coming?
You could see a little bit of what was going on with that animation. I think they're being very cautious as to whether or not they want to declare it, but Rob, what have you been hearing?
Yeah, we went into the winter really warm, beginning of the Bering Sea was sort of the center of the ridge activity. And that, 2-3 week sunny spell that we just ended, that's what led to Blob B1. And this year, that ridge was centered on the Bering. And they set a new record for loss of ice mass. This last warm epoch had lost over 400,000 square kilometers of ice. So the Bering Sea now looks like it should look in June. So yeah, quite warm.
Thank you guys for coming.